Good morning. First of all, today's lecture is going to be on Poisson random variables or Poisson processes and how they relate to genomics, biology, more focused on genomics, and in particular, both bulk RNA-seq and single-cell RNA-seq, specifically how Poisson random variables or Poisson randomness impacts the technical noise that we see in these experiments. Before we get going, though, I'm going to quickly go through how to use R Markdown within RStudio. I mentioned last time reproducibility and basically documenting your code, not only with saving it as a script, but you can use this thing called R Markdown, which I didn't get a chance to show you, to save the script in a way that the script actually generates a PDF or an HTML document with all the plots and everything and all your pros or comments about the commands in one document. And the reason I'm going to go through this is I'm going to assign homework 3 today. And what I want you to do is, this is primarily going to be working in R, but you're going to be using Unix and R, but the R portion of the homework, I want you to submit as a R Markdown document. So everyone can see my screen, I hope. It says I'm sharing. At least I got that right today. So the way you do this, and please chime in if you're not catching up or if you're not following me, we're using RStudio at this point. What you do is you go to File, New File, oops, and then R Markdown. Okay. In this case, I uninstalled R Markdown, so if you don't have it already, you'll see something like what I'm about to see, which is it telling me that it needs to install some required packages. So R Markdown requires all these different things to be able to generate, to sort of compile all of your code and the plots into either an HTML document or a PDF. So I'm going to do that. Hopefully this doesn't take forever, but you can see basically what installing packages looks like here. It's going out to the internet, grabbing a bunch of R packages that it doesn't have currently, and try to install them. How many of you have actually used R Markdown before, just so I have a sense? Anyone, can you raise your hand if you have? Got, looks like 1. All right, great. So this will be new. We've got a couple people, so this will be new to many of you. I really like this. R Markdown is super nice because essentially it can be used to draft your manuscript. If your plots are made in R, and you can also convert it to slide decks. It's really nice. Okay, so it installed all the packages, and now I've got this interface here where I need to give my R Markdown file a name. So I'm just going to call this my Markdown. You could probably come up with a better name. There are 3 different output formats, HTML and PDF and Word. I would recommend either HTML or PDF. And then there's a document type. So typically you're just creating a R Markdown document, which is really just the combination of all your code, the resulting plots from that code and the output of the commands, as well as your description of, why am I doing this? Sort of maybe you have an abstract of the whole analysis, bit of motivation, questions being asked, that kind of thing, sort of like a preamble to the whole analysis. And so I will do that, click OK. And now I get, all right, so this hopefully looks somewhat familiar. So this is an auto-generated template here that was just created for me. What's kind of silly about this to me is it gives it a title called My Markdown, but it doesn't actually name the file that. So I'm actually just going to first save this file as my markdown.rmd. So R stands for R, and then MD for Markdown. This is some default commands that are used to generate the document. You don't want to change that. This is an example. So there are sections that are generated. This markdown is kind of like simple HTML. When you use pound, this means that this is a new section. And so when the HTML document is generated, you'll see R markdown in this case as in large text. I'm just going to call this introduction. And I am going to just say something like, this is an example type. So I am just going to make a little bit of R code here. 6, 8, 10. And I'm just going to plot. So what's going on here? This is a code block. So basically, whenever you're using R code when you're embedding the R code in your document, you start out the R code with backtick, 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 and then in curly braces, you tell it what language the following code is going to be. R Markdown actually supports Python and other languages, but it's most of the time you're going to be using R. So you're saying the language is R. Here's the actual R code that you would typically type in the console or on in an R script, and then you have to close out the code with 3 more backticks. That's so our studio knows where your code block started and where it ends. And then we can have another section, which is my toy analysis. And again, it's a new code block, starts with backticks, 3 backticks, telling it R is the language. I'm plotting X and Y, okay? So this is a very basic thing, but you could, for your homework, for instance, have a different section for question 1, question 2, etc., with a description and your R code. Now here's the magic. I'm going to save this, and now we can actually run this. I haven't done this in a while, so up at the top, there's this dialog called knit. One of the packages that R Markdown installed, it's called knitr or knitter. And it's essentially, this is the package that takes this document, figures out where our R code is, where plots might be, and generates an HTML file. So let me just show you this. I probably have an error, but we'll see. Down here, it shows us that it's generating some stuffs. Hey, and check this out. Now it creates a fancier looking HTML document. You can save this to your desktop, and then with your browser, you can just open up that HTML file, and you can see the parallels here. So R Markdown, that's my title. Oops, that's my window. Okay, so that title matches the title, author, date, and now here's this code block, and it makes it look nice and pretty. And here's the normal text preceding it. And introduction, as you notice, this is a markdown header section. So that pound introduction gets generated into this. If you've ever written HTML, this is probably an H2 tag, I'm guessing, something like that. We could look at the code behind this if we wanted. My toy analysis, and now this is what I think is really cool. The plot command that you use is in a code block, but then what happens is R Markdown executes the code to generate that plot, inserts it into the HTML, so you have this living document of your commands and your analyses. So hopefully this is enough for you to appreciate why this is useful. It wrote this file to my home directory. There's a ton of documentation on this online. There's probably nice little 10-minute videos that you could YouTube and learn more about what to do here. But this, I think, gives you enough to have a sense of what you need to do for your homework. But the reason I'm having you submit your homework in R Markdown is that it's a really nice system for doing your own analyses and sharing your work with collaborators, with your PIs, with your colleagues in your lab. Okay.
Dr. Quinlan, I have a small question. So if you want to knit an R Markdown, do you have to save it in the same file from which you generated your data frame? Like suppose you're reading an Excel document in a folder, would you knit? Do you have to knit to the same folder for it to work?
No, you just need to set your R Markdown document needs to explicitly say where you're reading, say, the Excel document from. That's where you could use that set working directory, or when you're loading that file into a data frame, you could use the full path to that file.
Got it. Thank you.
Yep, no problem. All right, and then the last thing, I think there's a way, I've never used this, but with the publish button, I think you can host your own little R Markdown collection. So you can have a series of notebooks online. I've never tried that, but it might be something you're interested in. All right, I'm going to move on. Again, if any of this was confusing, go back through the video. I'll post this later today, and there's a ton of really good documentation about R Markdown online. I'm by no means an expert, but it's not terribly difficult to use once you follow, especially since they give you a template. All right, so now what we're going to do is switch over. Can you still see my slides in presentation mode?
Yeah.
Okay, good. Great. All right, so we're going to talk about Poisson processes. You've probably heard of the Poisson distribution or Poisson random variables, and many of you have maybe even thought about how Poisson randomness or Poisson noise affects processes in biology. I wanted to do I haven't done a dedicated lecture on this every year. I really wanted to focus on this early this year because I think randomness, especially in counting applications like RNA-seq and ChIP-seq, where essentially we're counting reads, and those reads that are aligned to the reference genome are a proxy for sort of the biological signal that we're looking at. I think it's important to understand these processes early on in the course. All right, so the Poisson random variable or the Poisson process is named after this famous French mathematician. His last name is Poisson. He was prolific. He sort of like Gauss. He published in lots of different topics, everything ranging from math, obviously, physics, astronomy. He's one of the lucky 72 whose names are on the Eiffel Tower. And he is the creator or sort of formalized the notion of a Poisson distribution, hence the name. And this is an incredibly useful distribution for modeling what we expect for rare, and I emphasize rare, events that we can count on our fingers. So how many times does a light bulb go out in our kitchen per year? That type of thing, or per decade. So it's not common, it's quite rare, and they're countable things. It's a discrete entity. So the Poisson distribution, as we'll talk about in a minute, is a discrete distribution or a discrete random variable. That's in contrast with the normal distribution, which we talked about before, which has an infinite number of values. So height, for instance, human height is a continuous random variable. For instance, my height is 73 inches, but actually you can measure anyone's height to an infinite precision, and that's why it's a continuous random variable. In contrast, things like the Poisson distribution, geometric, etc., are countable. We can count the entities or the possible values on our fingers. They're integers. All right. So digging into this a bit more, this is used to describe the distribution of rare events in a very large population, and emphasizing the rarity, and then in a large population, and meaning that there's lots of possible outcomes. So I think a nice example in the context of this course is that at any given, at any moment in time, let's imagine our liver, the liver inside of us. At any given time, there's billions of cells in our liver, and at any given time, there's a certain probability that a given cell in that large population of billions of cells in our liver acquires a mutation at a particular site in the genome. That's an extremely rare event among a massive population, and that population is the product of all the billions of cells plus all the billions of nucleotides present in each genome in each cell. So it's, trying to model this extremely rare thing on a massive collection of possible outcomes. And so in thinking about this, Poisson is appropriate because mutation acquisition is a rare event, and each mutation, and this is one of the key aspects of the Poisson process, is every mutation or every event that you're trying to model with a Poisson process is thought to be independent of one another. So what I mean by that is, if there's a mutation at position on chromosome 1 in one cell, not only is that mutation independent of the probability of a mutation in another cell, but it's independent of a possible mutation at other locations in the same genome in that cell in which the mutation occurred. Now, you could imagine scenarios where that may not be true, because imagine that a mutation occurs in one cell, and then what we're actually looking at as another cell is a daughter cell of the original. So it might have that same new allele, but it's not a mutation. It actually is in that daughter cell because the mutation occurred in the parent cell, and then there was cell division that propagated that mutation downward. So key things to keep in mind with Poisson is rarity. It's a rare event, large populations of cells or lots of RNA molecules in the context of RNA-seq, and that every event is independent of one another. All right. So we can use this concept to think about a so-called Poisson process, and we can use this as a sort of a thought experiment or an actual way to simulate what we might see when modeling or counting the number of occurrences that appear. And what we have to do to actually model that is have some sense of the average number of occurrences over time or in the population. So one example that I like to use is, let's say that over the last 100 years, seismologists all around the world have determined that there is one earthquake above Richter's 6.0 worldwide per month on average. And so how did they come to this? Well, this is just basically looking at data and tracking seismology around the world over the last 100 years. And so they know when every event occurred, they know what the magnitude was, so they can come up with this sort of average. And that average turns out to be the key parameter, the only parameter that we end up using to model Poisson processes. But let's think about some poor seismologist whose job is to track high magnitude earthquake events in the state of Utah. Many of you remember, I think did we have one above 6.0 last year? Or I think maybe it was around 5. This is right after COVID started. I don't remember what the magnitude was, but that was sort of the extra punch in the face after COVID. Let's imagine you're a person monitoring all the earthquake activity in Utah, and you sit there all day and watch the little plot of seismic activity, and in month 1 you saw nothing. nothing above 6.0. There might have been a 5.9, there might have been a 2.8, but there are 0 events in month 1, 0 events in month 2. But on the 3rd month, you actually had some interesting data. There were actually 2 events above 6.0, and 0 the next month, 1, and so on and so forth, until after 32 months, you've spent, so what, nearly 3 years of your life tracking 6.0 earthquakes. And what you can see is we could build a histogram from this. So most months, it looks like it was either 0 events or 1 event. Occasionally, there was more than 1 event, 2, 3, a couple 3s. So it turns out that this pattern of countable events, 0s and 1s and 2s, is exactly what would be predicted if the average is 1 earthquake per month worldwide. Okay. Now, the key thing here, and this is sort of complicated by the fact that you can have aftershocks in the case of earthquakes, but let's just think of the aftershocks not being counted. We're really only counting the original event. So the number of events, the number of earthquakes worldwide are thought to be independent. So they can be modeled by a Poisson process. If they're not independent, if there's some reason to believe that they're not independent, then the Poisson random variable or Poisson process is not appropriate to actually model the expected number of events, mutations, meiotic recombination events, that type of thing. If the assumption of independence is violated, you can't use Poisson. All right. So the beauty of the simplicity of the Poisson distribution is that there's just one parameter, and this parameter is typically called lambda. And that lambda is nothing more than the average occurrence of the event. So in this case, the lambda was 1. The thing that we're trying to model as a Poisson process is the number of earthquakes above 6.0 per month. So there's lots of months to sample from, and the average is 1. That average is the lambda. Okay, and this lambda expresses the probability of a given number of events occurring in some fixed interval of time, in this case a month, or space, in the case of a genome, if these events occur at a known constant rate. So here is an example of 3 different Poisson distributions with 3 different lambda values or different averages. So let's focus on the yellow one first. This is a lambda of 1. And this is exactly the Poisson distribution that would be used to model the number of earthquakes above 6.0 every month. And what you can see is that since it's a very rare event, we're saying it's, on average one occurs per month. The probability of 0 earthquakes and the probability of 1 earthquake are the same, roughly 0.375 or 37, if I remember correctly. After that, the probability of more than 1 event, say 2 or 3 or 4 or 5, it just falls off like a cliff. Because the average rate is so low, seeing lots of events in a particular month, lots of mutations in a particular cell, etc. If it's rare and very rare, like an average of 1, then we see this quick drop off. And if you go back to this slide, what I can tell you is I simulated this data such that the frequency of the 0s and the 1s is the same, and they represent exactly this proportion of events in that 32-month period. So if we increase the expected number of events to say 4, then what you can see, not surprisingly, is that, oops, let me get this out here. We can see that the probability of 0 events now is actually quite low because the average is 4. 1 event is more likely, 2 events is even more likely, 3 and 4 events are equally likely, and then it falls off quickly after there. So even with an average of 4, seeing 8, 9, or 10 events is extremely unlikely. And then with an average of 10, it starts to look kind of like a normal distribution. And one of the properties of the Poisson process is that when the lambda is large, it approximates a binomial distribution, which is kind of like the equivalent of a normal distribution, but for countable events. So it starts to have that bell shape. Okay, so let's talk a little bit about the math. There's no reason to memorize this equation. You can look it up every time, but the beauty of this distribution is you can model the probability of a certain number of events K happening. So if we wanted to figure out what is the exact probability of 7 earthquakes happening if the average is 1, then we can use this fairly simple equation. We raise Euler's constant to the negative of the lambda. So that's the average, e to the negative 1. And then we multiply that by 1 to the k, which is the number of events that we're trying to model. So in this case, 7, divided by whatever k is factorial. Again, you don't need to memorize this. The whole point is that you can just plug this right into R. So if I want to model the exact probability of 2 earthquakes in 1 month, given an average of 1, I raise, here's e to the negative 1 squared, so lambda to the second power, k is equal to 2, divided by 2 factorial. So this would be the R code to do this math. And what we find out is, if we plug that in, I won't bother to, you can if you want, that the probability of seeing 2 earthquakes in 1 month, if the average is 1, is 0.18. And if you look on this distribution, yep, there's k is on the x-axis here, there's k equal 2, and yes, the probability is 0.183, etc. Okay, so I never want to have to memorize that equation. I never could, quite honestly. Thankfully, there's a shortcut in R. And we talked before about generating random variables from certain distributions. So we did R norm to generate random variables from a normal distribution. Similarly, we can generate things from the Poisson distribution, and the D family of functions in R allows us to get the discrete probability from a particular distribution, given the exact discrete value that we're trying to estimate or measure, and the parameter. In this case, for Poisson, the parameters, the mean is 1. So we need to get it. What number are we trying to figure out on this distribution, and what is the lambda associated with the distribution? And obviously, we get the same result, and we don't have to memorize all this math. Any questions at this point? Oh, I see something in this chat. What does 5.8 mean, Sarah?
It was the magnitude of the earthquake.
Oh, thank you. Thank you. Oh, it was very close to 6. Sorry, it's only 5 minutes have passed and I totally forgot the context. All right, so let's move into a classic application of the Poisson process. So many of you are familiar with the Adelbrook experiment. And this whole experiment, first of all, it was a genius experiment. But the question that they were trying to get at, this is a picture of Adelbrook and Luria at Cold Spring Harbor. The question they were trying to get at was, there's this observation that bacteria would sometimes become resistant to phage and growing bacteria on plates. And so the question was, how does that come about? And the two hypotheses that they tried to test was, one, the process of being, or the act of being exposed to phage allowed or triggered the bacterial population to mutate and adapt to that phage introduction. Hypothesis two is that there's standing mutation in the bacterial population that, in the absence of phage, doesn't confer any real selective advantage, but in the presence of phage does. So so basically what they found, they made two hypotheses. Those are the two hypotheses. And they thought that if it was a standing mutation, what they called induced mutation that already exists in the bacterial population, then they would see a pattern of, random colonies that would be resistant on the plate. Sorry, this is the plate on the bottom. Now, however, if it was if the resistance arose as a consequence of the phage introduction, then you'd have these sort of clonal expansions of colonies on the plate. And this pattern on the left is consistent with a Poisson expectation of the number of colonies that you'd have on a plate that are resistant. And this is inconsistent with a Poisson process. So in other words, the whole point here is that using a Poisson expectation and actually doing this experiment over and over again, they concluded that the mutations arise in the absence of selections. They're already present, in other words, already present in the bacterial population, rather than being a response to selection by the introduction of the phage. The broader point being here, just like this was an early application as a thought experiment based upon Poisson random processes. Okay, so a couple more examples that I want to walk through. So consider some position in your genome. If you could trace its ancestry, that position of your genome, back over the last billion years, so every genome that preceded it, and it back and forth. And so in human, generation time is, let's say 20 years on average. You're going back a billion years divided by 20, that number of generations. So working back in time. If you could just pick one position in your genome of the 3.1 billion positions, what's the probability that you'd actually see no mutations at that site across a billion years, or a billion divided by 20 generations? What I really like about this, and this is used all the time in population genetics and modeling genome evolution across species, and it underlies a lot of the work done in phylogeny, is basically we can model the expected number of mutations as we can figure out the lambda. So remember, for Poisson, we always have to have this lambda, and we can figure that out by simply multiplying the amount of the mutation rate and the number of generations, the number of years. So the mutation rate times time, because essentially that is how the average number of mutations are going to arise. So in this case, I've chosen two convenient variables. So let's say the mutation rate is very low, say 10 to the negative 9th, but the time is very high. There's a billion year, or 10 billion years going on here. So if we multiply these two together, we get an average of 1. Therefore, we can directly, with this lambda, we can directly model the probability of seeing no mutations at the site that you chose in the genome to look at. And that's, oops, looks like I changed this, 0. So we can just plug this in. Given a lambda of 1, and we want to ask what's the probability of seeing 0 events, we can say we know for directly from the Poisson distribution that this is there's a 36.7% chance that that single site in the genome that we chose will have never seen a mutation all the way back for 1 billion years. It's also equally likely, I know this from this distribution here, when lambda is 1, it's equally likely that we will see 1 mutation across that 1 billion years at a particular site. Okay. All right, so let's move on. We can also use Poisson processes to model other rare events that maybe we care more about. I spend a lot of time watching Bundesliga and English Premier League, especially during COVID. This has been a savior for me. And I looked it up a couple years ago. In the 2018-19 season, there was an average of 0.11 red cards or bookings per game, so a rare event. And we can use that as a lambda. We can figure out the exact probability of seeing 0 red cards, which is most of the time, like well over 80-something%. And the probability of seeing 1 red card, and we very rarely see 2 or more red cards. And that's depicted in this plot over here on the right. Now, this plot is made with something called Ggplot. I'm not going to install that today. Or I'm not going to run this today, but this package Ggplot2 is installed on my system. The way you would install it on your RStudio, if you haven't already, is use install.packages. So this is the common command for installing new packages that you don't have. And then in quotes, you put the package name, and you apply that, supply that to the install.packages command. It will go out to the internet and try to find that package and install it for you. Once you do that, you can load this library. If you've installed it, it doesn't mean it's loaded in your current working environment, so you have to do that every time. And then this is just showing, I'm modeling the probability of the number of red cards from 0 to 10. So that generates a series of numbers from 0 to 10. And then I'm figuring out the exact probability of 0 events, 1 event, 2 event, all the way up to 10 events, given a lambda of 0.11. So let's plug these two in. So we get, it's I guess there's a 0.895 chance of 0 red cards in the English Premier League. That might be different in different leagues, but that's what it is there. So then I can create a data frame. This is something we haven't done before, I don't think. We can create a data frame directly from those 2 variables. So basically you say data frame and then give it the X values, 0 to 10, and then the probabilities associated with 0 through 10 events given that lambda. So we get a new data frame now. So let's check that out. Right, so we get it named, and then what's great is it gives us a header, so we can name these variables directly. And then all I'm doing is using Ggplot, which is complicated. It's much more complicated to generate plots with Ggplot than the standard plot command. I'm not going to expect you to work with Ggplot for this course, although on the next slide I'm going to show you a lecture and a video from Tom Sassani, who was in my lab a couple years ago. He gave a really nice tutorial on how to use Ggplot to make these fancier plots. I'm really not that big a fan of them, but they are widely used. All right, so the links for that are right here. This is from a interest group that we ran a couple years ago called SLOBs, or Salt Lake Lovers of Biostats. Tom did a really nice job with this, and the commands to learn Ggplot are here, and then there's a video of him walking you through them here. All right, similarly, we can generate histograms of the probability of the number of home runs in a baseball game. They're pretty rare. Typically there's 0, but more typically there's 1 home run per game. But in 2017, 1.26 home runs was a record. And this is my favorite baseball player ever, David Ortiz, who hit a lot of home runs. So it's the same setup here. The only difference is now we're just changing the lambda. Instead of 0.11, this is a more frequent occurrence. So you can see that 1 is more frequent than 0 as compared to what we saw here, where given a low lambda of 0.1, the most common event is 0 red cards or 0 events. All right, we'll skip this, but this is the last baseball example where no hitters are extremely rare. So this is modeling the average number of hits per game. So as you know, very infrequently are there 0 hits from a given team. All right, so let's move into something, now that we've gone on a sports tangent, let's come back to biology. And let's talk about something that my lab thinks about quite a bit, which is mutations in genomes. We've already given one example here. One of the things that's been done over the last decade, thanks to whole genome sequencing, is trying to understand how often mutations occur on sperm or egg, so-called germline mutations. And what we found, we as collectively as a genomics community, we found by sequencing families. So you sequence the DNA of mom, you sequence the DNA of dad, and you sequence the DNA of child. And what you're looking for are genetic variants that are in the child that are absent from mom or dad. And the inference is that those occurred on either the sperm or the egg that went on to form the zygote that became that child. And therefore, if they're specific to the germ lineage of either mom or dad, you do not see that mutation in the DNA that we sequence from mom or dad's blood, but we do see it in the child because it occurred in sperm or egg, formed the zygote, and then is present in every cell, including blood from the child. Okay, so what we have learned over the last decade is that there's an average. If we look at any one of us, let's say there's something like 30 people on this call right now, on average, if we were able to sequence the DNA of my parents and me, you, and all of your parents, and do this exact same experiment, we would see an average of 70 new so-called de novo mutations or germline mutations that are specific to us, but not in our, and they're not in our parents. Okay, so in terms of studying, the reason we're interested in germline mutation rate like this is many times these germline mutations lead to rare diseases and developmental disorders that have that are really interesting from a clinical perspective. We need to know where these mutations are, what genes they affect, so that therapies can be developed. But these 70 de novo or germline mutations that we see on average in my genome or your genome are typically not affecting genes. The human genome is massive. It's 3.1 billion base pairs haploid. Only 2% of that 3.1 billion base pairs actually encodes proteins. So only 2% of the genome, roughly, it's slightly less or slightly more, but a good estimate is 2% of the genome is actually protein coding exons. So if we want to figure out the expected number of de novo or germline mutations that arise in sperm or egg that actually affect coding sequence, we can take that 70.1 and multiply it by 0.02 or 2% to figure out that we expect, on average, every one of us to have 1.4 on average. Certainly my genome cannot have 1.4 mutations. It can only have 0 or 1 or 2 or et cetera. They're countable. But on average, across all of us, that's how we get to this lambda that is not an integer. So our expectation is that 1.4 germline mutations per individual for every one of us. The average will be 1.4 coding mutations. So if we do that, we can, again, I know I'm beating a dead horse here, but we can plug this in as a lambda and model the expected number of coding mutations per individual. So if we looked at everyone in this class, we would expect that about 25% of us actually had no coding mutations. Something like, I don't know, 38% of us had one coding mutation, and an unlucky few, probably me, have 5 or 6 or more coding mutations. So this is a so-called discrete a density. Well, in this case, it's a discrete distribution where we're getting the probability of each individual outcome, possible outcome. But if we wanted to figure out what's a trick that we could use to figure out what is the probability of having one or more de novo mutations? Anybody have a guess? One hint. Yeah, go ahead.
I was gonna say, do we just take the probability for 0 and subtract it from 1?
Exactly. So that's what that was Kevin. What Kevin's getting at is something called the complement rule. So the idea behind the complement rule is the total probability has to add up to 1. So I've only looked at 0 through 10, but you can imagine this going all the way out to infinity, an infinite number. And the sum of all those probabilities, which are vanishingly small as we get larger, have to add up to 1. So with this d plus function, we can get the exact probability of 0. So then we can take 1 minus that probability, and it gives us everything from this bar to infinity, the total probability of 1 or more. Okay, so let me just show you what that looks like. 1 minus. So the probability of 1 or more de novo mutations in any one of our genomes is about 0.75. Okay. All right. So the other thing we can think about is, what is the expected number of de novo mutation in a randomly chosen large chunks of the genome? So if we expect 70.1 de novo mutations per genome, what I'm getting at here is that we can then model the probability of any event being in a, say, large chunk of the genome by essentially multiplying that 70.1 by the proportion of the genome that 1 megabase chunk represents. So 3.1 megabases is what? 3,100 1 megabase chunks. I hope I did that math right. I think I did. Yes, I did. So therefore, the probability of seeing a mutation, the lambda there would be 70.1 times 1 over 3,100. 1 megabase over 3,100 megabases. And so the probability is actually quite low for any randomly chosen 1 megabase chunk. Okay. All right. So what we did a couple years ago that I had a lot of fun with was doing a direct application of this. And the reason we did this experiment is the experiment, first of all, was figuring out whether or not the number of chocolate chunks in a cookie it can be modeled as a Poisson random variable. So the point I'm getting at here is the point of this experiment was I've walked you through a bunch of scenarios where we know what lambda is and the data follow a Poisson distribution. Now, the connection to biology and studying things like RNA-seq or drop-seq or things like that is we know we're studying a random process, but the question is, can that process actually be modeled by a Poisson random variable or Poisson distribution, as opposed to some more complicated discrete random variable, like a negative binomial, if you've ever heard of that. And so we basically have to do a goodness of fit test. Ultimately, we want to know whether or not the data actually support the notion that it can be modeled by a Poisson variable. Because if so, it's actually quite nice, because as I hopefully showed you, Poisson distributions are very simple to work with and think about. But it doesn't mean that our data are actually, the technical noise in our system actually follow a Poisson distribution. So the technical noise in the system here is, the human intervention of stirring chocolate chip batter and putting the spoon in there and scooping it out and putting it on a baking sheet and put it in the oven. Is that human intervention, the mixing that we've done, does it randomize things enough such that the number of individual chocolate chip chunks that end up in a cookie is really can be modeled with a Poisson process? So I wish we could do this year. COVID and Zoom prevent that. But what we did was, with this class a couple of years ago, I made some cookies at home, handed them out, and then everybody took the cookie and counted the number of visible chocolate chip chunks. And what we did was we put it in this spreadsheet. I don't remember who these, I know that's me. So the cookie I got had 2 chunks. And I'm not going to go through all this, but you can see all these different chunks. Each student in the class took a look and put their chunks in. So the question now that we're going after for the next few minutes is, are these observations modeled well by a Poisson random variable? So what's the first, any guesses as to the first thing that we need to do to answer that question? These are observed data. What do we have to figure out to model what our expected data would be with the Poisson distribution? This is what we observe, but we are essentially comparing what we observed to what we expect for a Poisson distribution. Any guesses as to what we need to do first? Here's a hint. What's the parameter that the Poisson process, needs? Well, the parameter that it needs, yes, thank you. Sorry, I just saw a chat pop up. Right, so Patricia says we need to figure out lambda. So can you elaborate? What is lambda? So how do we figure out what lambda is? Okay, well, in this case, lambda is nothing more, well, in all cases for Poisson, lambda is nothing more than the average. It's meant to be the average, or the expectation, the expected number of things, right? So all we need to do to compute lambda is, let's see if I can figure this out. And yep, all right, average, just take the average of all these guys. So the average is 3.63 forever. That's our lambda. Okay, so let's just go into R here, and I'm going to do our trick. We're going to use this discrete probabilities from a Poisson process. I'm going to model. Let's go all the way out to like a really awesome cookie with 20 chunks, and we'll set the lambda equal to what we just observed. So this is what we'd expect. Let's actually make a plot of this. Can I do this? Let's see if I can figure this out in real time. color. No, type equal both maybe. Yeah, good enough, whatever. So given our expectation or our average expected number of events on average, this tells us that it's very unlikely to see, we're very unlikely to see a cookie with 0 chips. 10% of cookies should have 1. Most cookies should have 2, 3, or 4 chips. And I think that makes some intuitive sense given that the average is 3.6. And then, you know, it's really unlikely to get that bumper crop cookie with 20 chips. So good luck. It doesn't look like anyone got that. All right, so now what we have to do is compare this expected distribution that we just generated from the lambda to what we observed in our experiment. All right, so let's work through how to do that. All right, so the first thing we're going to do, I'm going to teach you a little trick, a new package that I am kind of obsessed with. I got to pick up the pace here, but we can do this. It's called data pasta, not pasta. And the reason it's called pasta is it allows you to copy and paste data from, say, a spreadsheet into a data frame in R. So the first thing that you all would need to do is install this package. All right, so install the packages, quote, data pasta, and then we're going to load that thing up. I'm just going to say, let's load that library up so we have it. Now, what you're going to do is go to this Google Sheets. So the link is back here. You can go to this Google Sheets, and this is what I think is just so cool. I can just copy and paste, copy both columns. So just block select from A1 all the way down to B23. Copy, and then you're going to go over to R. And I forget how to do this every time, but I will remember it. Oh yeah. So up at the top, there's a little thing called add-ins. Let me just make a new script first. And you can paste. So see this data pasta section? You can paste it as a data frame. And what it does is it auto-generates this code based upon the values that were in that spreadsheet. Probably looks a little cryptic, but now what I can say is, I think I called this num chunks. Yeah, called it chunks. I created a tibble, but that's basically just a fancy data frame. Just do data frame. Chunks equals data frame. I can select all this code and then use my command enter trick to run it. And then I can take a look at what it did for us and check that out. We've got this nice, clean, pretty data frame that we can use for analysis, generated by just copying and pasting from the spreadsheet. Right. That's really nice. So now we can use our table trick to look at the frequency of different chunks. And what we see is that we saw 2 cookies with 0 chunks, 1 with 2, 6 with 3, 7 with 4. There were no cookies with 1 chunk. There were 5 cookies with there were 4 cookies with 5 chunks, 2 cookies with 6 chunks. Okay, that's our observed. On the right is our expected. How do they compare? That's our question. So I'm going to use. If you don't have Ggplot, you won't be able to generate this plot right now, but I'm just going to show you what the bar plot of these data look like. And by the way, just as an aside, Ggplot, Cow stands for Klaus O. Wilke. He is the author of this package called Ggplot, eponymously named, I guess. And this allows you to get rid of some of the ugliness of the Ggplot, and it makes it a little bit prettier. But again, this is just a bar plot of what we see in the table, a graphical version of what we see in this table. Right. All right, so I want to now talk about goodness of fit. We're going to install another package. If you're not keeping up, don't worry about it. You can go back and do this afterwards. There's a really nice package called Visualizing Categorical Data, or VCD. I didn't know about this till 2 years ago. I want to point out this link here. This is a phenomenal textbook by Susan Holmes at Stanford and Wolfgang Huber, who used to be at Emble. I don't know where he is now. It's called Modern Statistics for Modern Biology. It's an incredibly well done book. It is all free online. You can download the whole thing. They have really great R code examples. It goes into Poisson random variables. It has applications for RNA-seq and lots of different things. If you want to learn more about stats beyond the scope of this course, which is fairly limited, this is a highly recommended book. In reading this is where I learned about this VCD package. So I'm going to install it and load it. All right, now the function that we're going to use from this VCD package is there's two. One's called goodness of fit, and another one called rootogram. So let's first talk about this. What this is saying is, hey, I've got this data. This is my observed data. I've got these num chunks. Let's just look at this again. These are our observed number of chunks from like 20 some students a couple of years ago. And what I want this function to say is, how well does this data vector of values fit a Poisson distribution? The first thing that this function does, as we just did, is it's got to figure out the expectation. So it looks at all these observed chunk values, computes lambda by just computing mean of that. So it first does this, literally. I haven't looked at the code for that function, but I can guarantee it does that. It then uses that to do what we just did, which is to compute the predicted discrete probabilities for different numbers of chunks, okay? And it analyzes those observed and expected values and loads it into this thing that I just call goodness of fit, or GF. We can then use this rootogram function. So let me do this. To generate the plot on the right. We're gonna spend a few minutes talking about this. See if I can make this look less ugly. All right. So what the heck is going on with this plot? This is a visual, not a quantitative. This is a visual assessment of the goodness of fit. And how it works is as follows. The red line and points are the expected values from a Poisson distribution given the lambda that we computed on our own and this goodness of fit function computed on its own. So ignore the red, the gray rectangles. Just focus on the red line for now. That fits the plot that I made a little bit ago, at least the first part of it, because they're only looking at the first few values. So what are they looking at? They go out to 6 values, so it's basically a rescaled version of like this part of the plot, okay? And I think that makes sense. Zeros infrequent. The y-axis scale is a little bit different because it's the square root of the frequency. And that's just the square root instead of the absolute frequency is used mainly just to make this visualization a little easier to interpret. So red is the expected, gray is the observed. So if observed matches expected perfectly, the gray bars will hang from the red dot exactly to y equals 0. So we can immediately tell that there are more, or there are less, sorry, there are less cookies with 0 chunks than predicted, and there are more cookies with 2 chunks than expected. Essentially, there are no cookies with 0 or with 1 chunk, but we expect that to be fairly common. So, visually, this is a clue that no, our observed data do not fit a Poisson distribution well. And this is because it really wasn't a random process, because the hidden fact here is that when I made these cookies, I made them with my kids, and we intentionally moved chips around with gloves, moved chips around on the dollops of batter before we cooked them. So we introduced a bit of non-randomness when making this. Okay, so if the data actually did fit a Poisson, the observed data did fit the Poisson well, you would see these rectangles hanging right to 0. So let's show what that might look like. So now what I'm going to do, instead of the observed being the number of chunks from cookies that we actually made and manipulated, I'm now going to simulate cookies. I'm going to simulate 1,000 cookies. And again, now we're using Poisson, but we're going back to this R function, which generates random variables, 1,000 random variables from a Poisson distribution with a lambda of 3. So the mean number of cookies, chunks in the 1,000 cookies that I'm going to simulate is 3. So there's 1,000 cookies that I just simulated, and there's some cookies with 0 and 3, and hey, the lucky one was 7. I'm going to save that as simulated chunks. I'm going to do a goodness of fit on that. Now, here's the logical distinction here is we know for a fact that these sim chunks were generated from a Poisson distribution. So the goodness of fit better be better. It should be a better fit than what we saw with the observed data, with the actual data. And here's what we see. So this is much close. All the rectangles are much closer to the 0 line than in this other analysis. Okay. Some are slightly above, slightly below. And this is a qualitative assessment of goodness of fit. Quantitative assessments of goodness of fit can be done with things like the empirical cumulative distribution function, which is talked about in that book. We're not going to cover this stuff in the course. There are other goodness of fit packages which give you a quantitative value. This is just to kind of whet your appetite as to how we actually go through an important step in our analyses, which is actually convincing ourselves that our data are well modeled by a particular distribution, because if we can be convinced of that, as in this case, it liberates us to apply specific statistical tests. For instance, if you measure the weights of a bunch of different mice from numerous litters and you see that those weights follow a normal distribution, they're well fit by a normal distribution, that's great, because then you're liberated to use things like z-scores and just use the normal distribution to measure the probability of seeing a mouse with a particular weight. If it doesn't fit that distribution, you have to A, understand why, but also B, think you need to be more clever in how you actually model your data and do your statistical analysis on it. Okay. I have 10 minutes, and I just wanted to go through one other application of Poisson processes in the context of single cell sequencing. And I might need to lean on my colleague John Chamberlain at the end here to explain something again for me, but I'm going to go through the basics. So single cell sequencing, one of the early advances for this was so-called drop-seq. And for those of you who aren't familiar with single cell sequencing, the idea is that you try to encapsulate a single cell in a single droplet, say a droplet of oil. And in that droplet, there will be beads which have barcodes on them that allow you to track the number of molecules, say RNA or DNA molecules, that came specifically from that single cell in that single droplet of oil. Now, there's two things that we want to avoid. We want to avoid droplets that just have barcodes in them, but no cells, because that basically is no useful information for studying single cell biology. And the other thing we want to avoid is droplets that have multiple cells in them. Because if there's multiple cells in them and we're trying to study single cells, our inferences that we make about expression or the DNA complement of a single cell are confounded by the fact that many of our droplets actually have multiple cells. And how do we know that? How do when we get our data out of the experiment, so single cell sequencing, you go through this process to encapsulate single cells, ideally single cells in a droplet. You take all the droplets that you think have mostly single cells, some have 0 cells, some have 1, some have more than 1. You do your DNA sequencing and you take all the data that come out, and it's sometimes difficult to detect the fact that, ooh, actually, this collection of droplets had more than 1 cell on it. So the way that this process works from a high level is depicted in this cartoon on the right. So essentially there's some microfluidics. So basically you're flowing beads through this channel from left to right, and you're flowing cells from left to right this way. These two channels, these two ski trails here run into the same intersection, and through this channel, oil is being flowed, droplets of oil, so that as the cell and the bead come together and hit a droplet, you ideally get this scenario right here, where a single cell, this green thing, and a single bead get encapsulated like that. Okay, this is what we want on the right. We don't want that. That's a droplet of oil with nothing in it. And we don't want a droplet of oil with, say, 4 cells or 2 cells. And here's what it looks like. This is a little movie of it happening, where basically, you're flowing from these 2 different channels. Here's the oil, and this is what you get. And if you squint, well, I won't even bother, but I can kind of see little gray bubbles in there that I don't know if they represent the cells or the beads. I would guess the cells, but you can see some of them don't have any beads, and I've caught 1 or 2 that look like they have more than 1 gray blob in them. So that's what it looks like in real life. So what we're looking for is an average. I mean, one thing we can control here is the aperture of those gates that flow cells in, and the gates that flow beads in, and the gates that flow oil in, so that we can sort of govern the probability, modulate the probability of an average of 1 cell making it into a droplet. So let's think about this. If we let's just simulate 2,000 cells. Let's say we want an average of 1 cell, a lambda. So it's a rare event in a large population of cells. Let's say we want an average of 1 cell per droplet. Well, that means that we're going to get roughly 37.5 cell droplets with 0 cells, 37.5% of droplets with 1 cell, but then we're going to have lots of doublets, triplets, quadruplets, and that's really not what we want. So if we wanted to sort of push the envelope, if we wanted to shift things so that we tolerate droplets without cells in the interest of avoiding the worst case scenario probably, which is cell droplets with multiple cells, we could, maybe reduce the aperture of the cells going through, so cells go through more slowly, so it's more likely that just one cell pops in at a given time. And that would lead to obviously many more droplets with no cells, but it reduces the number of droplets with 2 cells or 3 cells or 4 cells. However, there's a tug of war here, because that also means that the efficiency of the single cell sequencing infrastructure here is less efficient, right? You're maybe spending more money, same money for less cells. And we could really ramp down that, close up that aperture, or lower the pressure or something like that. I don't know what the engineers do, but you can imagine things that you might do, like kink the hose or something, and really slow down the number of cells such that you basically only get 2 scenarios: droplets with no cells or droplets with just 1 cell. And that's by really lowering this expectation through mechanical means, probably. The point here is that a priori, if you think about this, the probability, if you have to do this you have to pull these tricks out to minimize the number of cells that end up in a particular droplet, the probabilities get pretty low. And then that's just for cells. Remember, we had this other gate, which is getting the droplets. We want 1 droplet per, 1 bead per droplet as well. So that also follows the Poisson process. And so we end up with this sort of pessimistic scenario where if we have to reduce lambda to 0.1 to get ideally 1 cell per droplet, and do the same thing to get 1 bead per droplet, then the probability, the naive probability, this we're gonna have to lean on John if he's around in a second. The naive probability is that 8 out of 1,000 droplets, oil droplets, will have exactly 1 cell and exactly 1 bead. Now that's a poor bit. That seems to me like that's a poor business model. And thankfully, these systems are doing a better job. And John has explained to me like 5 times how they do this. John, can you jump in and help me say why it isn't this bad? If you're on. If you're not on, that's okay. So it looks like John isn't on today, so I will follow up why that isn't the case. It's primarily, there's something about the system that they how they modulate the number of cells and droplets that go in to maximize, to get this well above 8 out of 1,000 cells. Okay, I'm going to stop here. We're going to transition next time to a lecture on the fundamentals of RNA-seq, and we're going to talk about jumping in. We're going to jump into Poisson processes in bulk RNA-seq and how it affects things like differential expression. In the last minute, I just want to highlight that there's a new homework assignment. It can be found here. It's also up on the GitHub site. It's due a week from today at midnight. And what I want you to do is highlighted here, is to submit it as an R Markdown file. So go back to this lecture once I post the video. There's also great material online. The last thing I want to say, the point of this homework, just to give a big picture, is to get you attuned to the process of working with data files on Unix, scrubbing them, getting them to a near final state, and then loading them into R and doing some new analyses and honing your skills and working with data frames, profiling data and data frames, and making some informative plots in the context of RNA-seq. Okay. Any questions at this point? Okay. Well, reach out if you have any, and I will post the lecture later today. All right, take care, everyone.
Thank you.